Hi everyone, it's Christina Wallace and this is Biology 233, Human Anatomy Physiology Lab, and today we're going to be talking about exercises 42 and 43, the reproductive system. Okay, so let's start right here at the online instructions. Once again, Professor Patterson has done a really lovely job of creating or giving you links to materials. And so the first part of your lab report starts out on page 1 with male reproductive structures, and there is a video right here that goes over a model of male reproductive structures that I would get familiar with, so be able to identify things on a picture and identify things on this model. And then on page 2, we're going to start looking at some histology. So I'm going to go over a few things on this instructions page, and then I'll go to the lab. So I just want to show you a couple of things. Histology guide, I'm going to show you a bunch of still pictures that I took from histologyguide.com. So you are free to click on that link and click on the vocabulary on the link. Because it will zoom into stuff. It doesn't put arrows next to things, but it will zoom in on stuff for you. And so just keep a look at this picture because it's a very useful picture and good to compare to pictures, images that you see on my slides, and images you might see in the book because everything is a little bit slightly different. And so this picture is a seminiferous tubule. And so a seminiferous tubule is a tube full of polka dots. You know, you're going to have to tell two different kinds of tubes apart in the male reproductive tract. So seminiferous tubule is one, and it's the one that has all the polka dots in it. Polka dots, big polka dots, little polka dots, big cells, little cells, etc. And so here is kind of a range of all the cells that you will see. I draw your attention to another Sertoli cell. I think this one right here is really good because it looks kind of like an avocado or an oval with the pit in it. That's a Sertoli cell right there. That's what you're looking for. And it's basically the nucleus of a big cell with a nucleolus inside. And the cytoplasm of the cell we can't see very clearly. And then the instructions, you're going to look at a histology guide of an epididymis, and then you're going to look at a cross-section of a penis on the histology guide, and I have still pictures of those things. So here we are in your lab report. I've given you a couple of examples about how to write functions. And so it says briefly, tell me briefly about the function. So you don't need to write big paragraphs here. And so I have given you a couple of examples of what a brief functional definition for these structures are. All right, so then on the second page of your lab report, we'll be looking at slides of seminiferous tubules. And so I've given you some information here. And then they're going to ask you functions of sustentacular cells. These are also called Sertoli cells or nurse cells sometimes, but you can find that information in your lecture notes and in the textbook. So let's look at some seminiferous tubules. These are the things that you're going to see inside of the tubes. So all these different kinds of cells are going to live in the tubes. And then in between the tubes, outside of the tubes, we're going to see interstitial cells, interstitial, in between, in the interstitial areas, like where interstitial fluids would be. And these are going to secrete testosterone. Interstitial cells do. And then all of these cells that you see in the tubes themselves, spermatogonia, spermatocytes, spermatids, and sperm, and the avocado-like Sertoli cells, these are basically going from the back of the tube towards the lumen. And so we're always going to be headed towards the lumen of the tube as our sperms mature through their cellular process. Another name for sustentacular cells or Sertoli cells are stenocytes. And so you'll see that term in your book as well. They help sustain the spermatogonia and spermatocytes on their journey towards the lumen. They nurse them along. That's their job. Okay, so here's a seminiferous tubule. It's kind of a funky shape, but that's okay. Spermatogonia are going to be at the very farthest away from the lumen, right against the edge of the basement membrane. The green arrows indicate spermatogonia. Spermatocytes are both primary and secondary types. It's kind of like, is it a 2N structure still, or has it split in with meiosis 1 to become a 1N structure? And those cells are all going to be found basically in the middle of the tube somehow. So spermatocytes. And I'm not differentiating spermatocyte. I'm not differentiating between primary and secondary, and you won't have to either. But they're the ones that are in the middle. So in the middle of the tube, of the cells in the tube between the basement membrane and the lumen, that's your location for spermatocytes. Spermatids, we are 1N, we're developing, we're shedding our extra cytoplasm now, and we're on our way to becoming a sperm in the lumen. Spermatid is the last stage, and those are going to be small and round, and we're going to find them at the lumen. So all of these circles, smaller, round, dense circles close to the lumen are spermatids. And here we are looking at Sertoli cells. And so the thing I want to explain about Sertoli cells, the textbook picture I think does a really good job. And so when you're looking at a Sertoli cell, like I'm going to circle a Sertoli cell nucleus. There's a Sertoli cell nucleus. This arrow is pointing at another one. There's a Sertoli cell nucleus. And you can see that tiny little dot of a nucleolus inside of it. What you're seeing is this structure right here with a little dot in it. You're seeing the nucleus of the cell. But then the cytoplasm of the cell extends all the way up to the edges. And so this is the cytoplasm, basically, of the nurse cell. And so as the spermatogonia, so spermatogonia on the edge of the membrane, right? And it's going to basically traverse through the cytoplasm of these nurse cells towards the lumen. And so you'll have primary and secondary spermatocytes in the middle, and then you're going to get early and late spermatids out towards the lumen, and then once you get in the lumen, you are a sperm. And so I just want you to remember this, this is the nucleus, the little dot you would see is the nucleolus, and the cytoplasm of that cell is big. That's a sustentacular cell. That's a sustenocyte or a Sertoli cell. You could use any of those names and it would be fine with me. And so I've pointed out a few of them here in this more zoomed out picture. So you're looking for anything really that has kind of an oval shape and a polka dot in the middle. So all of these are Sertoli cells. Here's one over there. That's a Sertoli cell. That looks like a nucleus of a Sertoli cell. And I'm going to zoom in and show you a better picture. So here is a much more zoomed in picture of sustentacular cells. And so again, the green arrows are pointing at these little cells with an avocado pit on the inside. This cell right there. That's the nucleus of a Sertoli cell, and its cytoplasm would go all the way up like this. Does that make sense? Okay. Interstitial cells now don't live in the tubes. Now we are between the tubes or outside of the tubes, and they're going to be in these kind of triangular or areas outside of the tube. And so any cells that you see in those spaces between the tubes, so this is a tube right there, and then this is a tube right there, and then there's a little piece of a tube right there. So we're in the middle, in the interstitial space, and that's where you'll find interstitial cells. And so that's pretty much page 2 of your lab. And so now we're going to go look at the epididymis. And so where the seminiferous tubules were tubes full of polka dots, the epididymis is going to look very different. It's going to be neat and tidy tubes with really clearly defined cell edges of their epithelium at the lumen. Like it looks like a little long line drawn around the epithelium at the lumen. It's very clear looking. It's not all messy with all those polka dots going up into the luminal space. And so this is what the epididymis looks like. This is the comma-shaped organ off to the side of the testis. The testis is where the seminiferous tubules live, and the epididymis is the little comma structure off to the side. We can see that, we can see that in this slide. So this is the testis, and then this is the epididymis. So this is the epididymis. And that's what we're going to look at in a cross section now. So you can see very clean edge to the pseudostratified columnar epithelium. And so here you have your columnar cells with their nuclei kind of at the base. And we do have some cilia here, but they're not motile cilia. They're called stereocilia. All of these fuzzy things right there. And they help reabsorb fluids from the tubes and reabsorb sperm that don't make it. And help to bring nutrients to the sperm. And so we can see some sperm like right here. This stuff right here, that's all sperm inside of a tube. That's sperm inside of the tube. And I'll zoom out to a better picture that shows more. This is epididymis. Very different looking from this. No polka dots in the epididymis. Here's another picture from histologyguide.com about the epididymis. And so you can see the pseudostratified columnar cells with their kind of odd nuclei at the base, right? And then you can see the stereocilia. These yellow arrows are pointing at the stereocilia. And then you can see all of the sperm in the middle of the tubes. So nice, clearly defined tubes with a clearly defined line at the lumen. Very different from a seminiferous tubule. Here's a cross-section of the penis. The penis has 2 large corpora cavernosa, singular, corpus cavernosum or corpus cavernosus. Don't worry too much about spelling on those. And these are the large areas. This right here and this right here, those are corpora cavernosa, and they are fed by deep arteries in the middle, and all of these little spaces would get filled up with blood during an erection, and then this membrane on the outside would keep them kind of firm so that the penis elongates rather than gets really wide. So that it can better deliver its DNA material into the closest to the uterus. And then the corpus spongiosum is another body of erectile tissue. There's only one of them, so there's no plural name for that. And it is this one right here. And the urethra is going to be the opening on the inside. So this little funky shaped thing, that's the urethra. And we're going to see transitional epithelium in the urethra. And then I just have, I have this picture labeled with some other pieces of architecture that you need to know. So here in your lab report, we have found all of those structures. I labeled everything on your pictures, so it should be pretty easy to find stuff. The one thing that I had difficulty finding in your book is the types of epithelium that line your urethra. And so I just want to give you these answers below. Transitional epithelium allows for stretch, so volumes of urine can pass through those tubes. Pseudostratified columnar tissue is all about secretion, and so we want to secrete lubrication for the urethra when the seminal fluids go through. We want to secrete some alkaline products to neutralize any leftover urine in the urethra. So pseudostratified columnar is all about secretion mostly, and a little bit about protection. And then stratified squamous, when we exit and we have bunches of layers, that's all about protecting from wear and tear and protecting from irritation. We have those layers of cells that can be sloughed off to protect. Okay, now we are going to look at the female reproductive system. And so I've given you a couple of examples of brief definitions of functions. You don't need to write paragraphs here. Just tell me basically what these structures do. And then back at the online instructions, you have a video here that will go over the model of the female reproductive structures, just like we did the male. And then there is also another video going over the parts of the ovary. And so I think this is a really great video. It goes into a little bit more detail than you need, but they are going to walk you through the ovary. So I'm not going to spend a ton of time walking you through the ovary because you have this great video to do that. Then we're going to look at the structures of the ovary. Once you have ovulated an egg, the follicle that is left behind becomes a scar. And so we're going to look at what those scars look like. And you have a hormonally active version of the scar, and then you have one, once it's done being hormonally active, it's just a dead tissue scar made of connective tissue, and we'll see both of those. But corpus luteum is the active one, and I will point that out to you on a slide. And then histologically, we're going to look at the layers of the endometrium during the menstrual cycle. And so we're going to be telling these different regions apart from each other with some characteristics. And I have some slide pictures, still pictures from Histology Guide, and I'll walk you through that stuff. So the first thing that we're going to look at on the female reproductive histology is an ovary. And so here's all the structures that we're going to identify there. We're going to find the germinal epithelium is often called surface epithelium, and the video about the ovary talks about why early histologists named it germinal epithelium, and so it's a pretty cool story. And then we're going to look at some different types of follicles. So you have a primordial follicle, a primary follicle, a secondary follicle, and a mature or Graafian follicle. And then once we get to Graafian, that's where we're going to ovulate an egg, and it's going to go into the uterine tube and make its way towards the uterus. And then there's some specific structures of a mature follicle that we're going to look at. And then the active scar left over is corpus luteum. So here's our ovary. It's really cool. Ovary has a cortex and a medulla. And so this middle area right here is pretty much the medulla, and you'd see the ovarian ligament kind of coming in to anchor it. So there's a bunch of connective tissue in there. And then there's going to be a bunch of arteries and veins hanging out in there where the blood supply and the hormone supply comes from, or where the blood supply comes from, I will say. And then on the outside, this is all the cortex, this area. And so follicles are going to mature in the cortex. And then the germinal epithelium would be right on the very outside, and we'll zoom in and take a look at that, or the surface epithelium. It's kind of like the visceral serosa or the visceral peritoneum of the ovary. All right, so we've zoomed in on the cortex. You can see a tiny, tiny piece of the surface epithelium right there, those little kind of cuboidally cells right there. little cuboidal cells right there. So that's surface epithelium or germinal epithelium. And then primordial follicles are basically an oocyte with one single layer of simple squamous looking cells around them. And they tend to gather towards the outer edge of the ovary. And so we're really close to the edge of the ovary here. And so all of these structures that I'm circling are primordial follicles. And they hook up in an egg nest. Like this little group of them is called an egg nest, which is just kind of cool. And so a primordial follicle, you can kind of see it just has these little flat cells around it. One single layer of little flat cells. So there's a nucleus of a cell, nucleus, like there, little flat cells. And so inside you have an oocyte, and then you have a nucleus of the oocyte. And we don't have anything in between the single layer of squamous cells. These are called pregranulosa cells. If you called them granulosa cells, I'd be fine with that. So that's primordial. They group together close to the edges. Our next stage of follicle is going to be a primary follicle. And so that's what we have right here. My pen, right here. That's a primary. So this one right down here is a primary follicle. And so you can kind of see the oocyte on the inside and the nucleus of the oocyte. I just did in yellow. And then on the outside, you see all of these layers, though that single layer of squamous cells has now become a single layer of cuboidal cells. And so that's pretty much our tell for a primary follicle. Single layer of cuboidal cells around an oocyte. All right, so a secondary follicle, we're getting closer, we're getting more mature on our way to ovulation. And so a secondary follicle, that single layer of cuboidal cells, has now become a stratified layer. There's multiple layers of cuboidal granulosa cells. So that's all of these cells right here, granulosa cells. And then we can still see our oocyte in the middle with its nucleus on the inside. And then we're going to start seeing what's definitive about a secondary follicle is this liquid-filled space right here that I'm coloring in yellow. That's called antrum, or the antrum cavity. And so basically we're going to start separating this oocyte and some cells from the outer layer. So we need to separate the yellow from the green. And so we're going to start filling that follicle with liquid as it swells. The corona radiata is this crown of cells that stays around the oocyte. That's corona radiata. And then this space on the inside, you don't have to know what it is, but this space on the inside that looks like it's filled with liquid, that's called the zona pellucida, and it's a glycoprotein substance, and that's what controls the ability of one sperm only to fertilize this egg. So once we get one sperm that breaks through, here's my little sperm and its little tail, breaks through that blue zona pellucida, it sends a chemical reaction through the zona pellucida that doesn't allow any more sperm in. So the blue area, zona pellucida, the green area, corona radiata, and the oocyte is in the middle. And the liquid-filled space with the aqua arrow is antrum, and then all the outer cells that make up the outer part of the follicle are granulosa cells. So now looking at a Graafian follicle, this is our most mature stage of follicle, and it's pretty much ready to be ovulated. And so, again, it has a big antrum cavity in the middle, antrum cavity. It has the blue, oh no, it was green, I believe. The green corona radiata going around. Here's another corona radiata over here in between the black arrows. Here's a secondary follicle over here. We can tell that because it's got a little bit of antrum forming. And then let's see. The granulosa cells are on the outside. Theca or thecal cells wrap around the follicle, and they provide hormones to the follicle to help it develop. And so it's these kind of cells that wrap around right there. There's like theca interna, and then there's theca externa. Well, I did interna in yellow, externa in green. But there are these cells that kind of wrap around the outside, right around the outside of the follicle. Those are theca. And so this is what a follicle looks like when it has already ovulated an egg. And so here I'm outlining in yellow this antrum cavity. And so in here would have been our oocyte with its nucleus and its little, it was green, I'm going to stay with the same color, with its little corona radiata of cells, little cloud of cells around it. That would have been ovulated out of the ovary and into the uterine tube. So this hormonally active tissue, so here, these are the old follicle cells. So that's the rest of our follicle once we have ovulated an egg. And so all of this stuff right in here that looks kind of bumpy and has these little lines and stuff, that is a corpus luteum. This is the hormonally active scar of a follicle left behind, and it secretes progesterone, some estrogen, but mostly progesterone, and it will keep the uterine lining healthy if a fertilization has occurred until the placenta can take over. And if no fertilization occurs, then it will stop being hormonally active in about 10 days, and it will become a corpus albicans, which is this structure over here in yellow. And this is just the scar left over. It's not hormonally active anymore. So corpus luteum here, corpus albicans, white, dense irregular connective tissue, non-hormonally active. Corpus luteum, hormonally active. And then looking at the lining of the menstrual, of the endometrium. And so if you're looking at the menstrual phase of endometrium, you can bet there's going to be kind of some shredded appearance up here, right? You might see some red blood cells in the area. We're shedding the endometrial lining, so it's going to look kind of torn, and there's going to be dead cells and stuff, and this is the beginning of the menstrual cycle. So we've just gotten rid of all of our endometrium, and we need to grow it back. And so that's what proliferative phase is. We're increasing the size of our endometrium, getting it ready, kind of making fertile ground again for a possible fertilized egg. So your endometrium has a couple of layers. It has a kind of a base layer, the layer that is closest to the muscle. So the muscle is down, sorry, I wanted to change colors. So the muscle is down here. Then the yellow is the base layer or basal layer. And then all of the stuff above that is the functional layer. So the functional layer is the one that's going to grow and shed. And so proliferative phase, this does look pretty wide, but the glands are more or less kind of straight, and they're just beginning to form. They're not all zigzaggy and giant yet. So that's indicative of proliferative phase. Endometrial glands are developing, but they're basically kind of more straight. And then secretory phase, this is the stage when ovulation occurs. And so this is the fertile ground ready for an egg to be implanted. And so You can see it's indicated by very exaggerated endometrial glands. And so all of these zigzaggy things, zigzaggy, all of these are glands. And they develop and really get big and active during the secretory phase. And it's the thickest of the 3 layers because it's fertile ground. We're ready for an egg to be implanted here. And so that's this last part of your lab. Just kind of know how to tell those 3 tissues apart. And then we're moving on to the physiology of reproductive hormones. And so now we're going to be kind of looking more at hormones. And I think you can, the rat questions of this, you're just basically going to be looking at the estrous cycles of the rat and trying to figure out how it correlates to human menstrual cycles. On the next page is where you see information about the corpus luteum. Corpus luteum. And I just want to draw your attention to a couple of places where lecture notes are useful. So on this, when you're talking about the function of estrogen, your lecture notes in the repro section, page 24 and 25, are useful. And then the last 2 questions of your lab ask you to paraphrase the negative feedback cycles. And so I'm going to show you, I'm going to go over a little bit the areas in your textbook. I'm going to show you textbook pictures and talk about that a little bit. But you can also find information in your lecture notes. So the male negative feedback cycle is on page 10. And the female negative feedback cycle in your lecture notes is on page 23, page 23. And those are the textbook pages that I'm going to talk about. So first of all, just a reminder, this is a great picture out of your lab manual and your book, and it just kind of shows you the color-coded. So the blue is the menstrual cycle here. Erase that. And then the proliferative phase here. And then the secretory phase there. And so it shows you where ovulation occurs. It shows you where corpus luteum forms. It shows you the fluctuation of estrogen and progesterone. And it shows you the layers of the endometrium and what's happening with them. So I think this is a really great overview graphic. And so this is pictured from your textbook. I've indicated the page numbers here. And so this is your feedback cycle. This is what you need to be able to articulate on an exam or tell me what happens. And so you're starting at the hypothalamus and you think about what gets secreted from your hypothalamus to tell the downstream structures what to do. And so it's going to release GnRH. GnRH is going to go to our anterior pituitary. It's going to cause it to release FSH and luteinizing hormone, follicular stimulating hormone, FSH, luteinizing hormone, LH. Follicular stimulating hormone is going to go tell the follicles to mature, luteinizing hormone, all of that stuff. And so once we get to increased level of estrogen in the blood, we'll start inhibiting and saying, hey, we don't need any more FSH anymore. And so this is kind of the beginning of the menstrual cycle, and then this is later in the menstrual cycle. And so your luteinizing hormone surges before ovulation. Ovulation is going to lead to a corpus luteum, which is going to secrete progesterone. Once we start secreting progesterone, we'll go tell the hypothalamus, hey, we don't need any more GnRH, we're good to go right now. And so just kind of make note of the keys and be able to walk through a little bit of this hormone negative feedback loop. What stops hormones from being secreted and what starts hormones from being secreted. What stops hormones from being secreted is the negative feedback loop. That's what you're looking at. All right, this is a horribly long video. It's my last one though, so thank you for listening, and happy studying to you.